Good evening, everyone. Welcome to Flyway Nights. I'm really excited to be sharing with you this evening about our fellowship program. And it is a little sad for us in that this is the last time we're going to be hearing from the fellows. It's our last year. We've been doing this program since 2015. The program was developed by our founder, Robin Kaleko, and to encourage students to do research, in particular around the Yolo Bypass wildlife area and wetlands. And so this evening, we're going to be hearing from the last of those four. I'm going to share with you my screen so that you can see who those people are. And this evening is going to be our Yolo Basin Foundation Graduate Student Fellowship recipients. And we will be hearing from our four recipients one by one about their various projects. Next month, we're going to be hearing from our own executive director, Martha Diepenbrock, who will be sharing with us about an exciting opportunity and grant that we're involved in for public access and education and capital improvement project for the Yolo Bypass wildlife area. And she'll be sharing about what all that means and also looking for some input as we begin to develop that program, which is going to take us over the course of the next couple of. So bring you back, and I'm going to now pass on to our fellows. All right. So again, let me share with you our students that we're going to be hearing from this evening, some of which have now moved on to being students. So this is the order we're going to be going through to learn about the various projects. There is Sara. Take it away.
All right. I hope the screen is up good.
It is. Thank you.
Perfect. And I'm going to be looking like a bunch of different places because I have two monitors, so don't mind me looking like a chicken with my head loose. Just try to pay attention to the presentation. So, hello, my name is Sara. When I started this project, I was a master's student in environmental policy and management at UC Davis. I have since graduated and I have moved on to the East Coast now. I am getting my PhD in mass communication and life sciences. I focus on environmental issues and climate change. So I've kept my niche, but I've switched a little bit field-wise. So with this presentation, I'm going to go over some brief background and introduction and methodology of what my project was. Then we're going to go over some results, and we're going to do a tiny bit of discussion over what these results can mean for the future. So we're going to start with our background. So nature preserves and wetlands have provided a lot of benefits to humans. They provide recreational spaces, research and education spaces, resource banking benefits. And in California, specifically in the Central Valley, a lot of these preserves are wetlands. They provide a lot of flora and fauna, especially endangered ones. They provide water filtration. They provide flood mitigation, and then they provide, of course, as well, food and shelter to many people who are indigenous to the land and have lived here since time immemorial. For Yolo Basin specifically, that would be the Patwin people. But even with its many benefits, there's still literature that suggests that there's increasing negative perceptions of wetlands as we age. So as we get older, we like wetlands less. Probably has something to do with the more present negative depictions of wetlands in media, literature, and fiction. I grew up with the constant fear of getting swallowed by marshes. That did not turn out to be um, as important of a fear as I thought it was. I thought it was going to be when I was like 12. So thankfully, there's also ever increasing calls for doing more environmentally conscious school curriculum in the United States. It has a lot to do with the rise of climate change. And of course, there are many NGOs that are focused on outdoor education programs and field trips. And one of those programs is Yolo Basin Foundation. Yolo Basin Foundation's largest program is actually Discover the Flyway, which provides field trips and in-class activities through K through 12 students. It is based on California Science and Social Studies standards. And their stated goal is to foster healthy outdoor experiences, develop land stewardship ethics, and promote stewardship of wetlands, agriculture, wildlife, and all that good stuff in the Central Valley area. So for my project, the goals was to examine to see if Yolo Basin Foundation is actually succeeding in reaching these goals. And I was going to do that by assessing the experiences of teachers and students that were involved in these field trip programs. So we're going to look at methodology very quickly. This is a pilot evaluation. So I looked at the program from 3 different angles to try to figure out, get a really big, wide, open view of everything. 2 of them focus on student participants. One is simply looking at the students' knowledge acquisition. Are they learning, and then are they remembering the information that they learned? And then another aspect is looking at the students' attitudes towards outdoor environmental education. How do they feel about science and nature after field trips? And then the last angle is focused on teachers and utilizing semi-structured interviews. Semi-structured interviews are basically interviews with some prepped questions, but they leave room for the interviewer, in this case me, to go off script and explore other subjects if they are brought up by the participants, in this case, the teachers. I choose to interview the teachers for 2 specific reasons. One, I count the teacher experience and teacher attitudes about the program as a crucial part of the student experience and them being successful. So if the teacher is happy and enthusiastic, and gets excited about field trips, then their students are more likely to participate and engage. And also the teacher is more likely to come back and participate every year in the field trip programs. And then the next part is it's hard to interview individual students, especially because they are very young. In this case, the students that I surveyed were between 3rd and 5th grade. So they have shorter attention spans. So interviewing with the teachers can provide me with the opportunity to ask them about any changes that they've observed in their students, instead of having to interview every single student, which would not be feasible given the time constraints of one such projects. So the project timeline, most of my time in fall of 2022 was spent prepping and meeting with Heidi, making sure that I am designing an experiment that is productive for the program, doing literature review, designing the survey, and the instruments for the students. So most evaluations rely on pre-post strategies to evaluate a program. That basically just means looking at performance before they go on the field trip and then looking at the performance after they go on a field trip. Looking at a lot of the literature, I decided to do a pre-post strategy of evaluation that just basically adds a second post a significant amount of time after the first post. So in this case, it would be about 2 months after their field trip. And the idea is to see if the students have retained information long term. Do they just remember information right after the field trip, or do they remember and have attitude changes long term? And then part 2 in the timeline is actually doing the surveys and interviews, this was very challenging. I had a goal to get 3 to 5 classes to volunteer. And it turns out it's really hard to get teachers to agree to set aside 3 30-minute chunks of their time and their students' times for stuff like this. So I successfully recruited 3 classes, which was very hard to do. And in total, that means that there were 114 students that were surveyed for this. And then we get to data cleanup and analysis. This part was also a little bit tedious. I had to look through all the interviews and all the surveys, drop all the students that didn't finish their surveys, and also drop students that were absent for one of the 3 surveys, because then that would just mess up the analysis. So doing all of that, I am left with about 70 students that have finished all 3 surveys. It's pretty good. And then in addition to that, I have to transcribe code and do all the fancy stuff with the teacher interviews. And for the analysis and visualization, just to be safe, I recruited the help of the University of Wisconsin Madison Survey Center, just to make sure that there was a second eye on the statistics. And then report writing, this is where I am right now. I have all the data, I have all the visuals and graphs, and I'm working on finalizing the report, going over all of it, tying it up, and putting a nice little bow on top of it. So let's look at some results. So we're going to start with the student results, with the good news, which is the knowledge part. It seems that in general, there is an increase in how well students are learning, retaining, and then also recalling information. So they are separated by class because not each of these classes had the same stations. Therefore, the questions that they had to answer would be different. So I couldn't pull them all together because that wouldn't be fair. But it seems like in general, so if you look at these colors, low just means there were 6 questions. Low just means they didn't get many of them right. Medium means they got a good amount of them right. And high means they got most of them right. And we have the first survey, which is taken before their field trip. The second survey is taken right after their field trip. And the third survey is taken 2 months after their field trip. So during the first survey, you can see our class C is just generally a very strong class. So they are a little bit of an outlier, but in general, you can see it's in low and mediums, and then we gain more knowledge as we go. And it seems like they are retaining a lot of their knowledge. They do lose some knowledge sometimes, but they are not like reverting 100% back to not knowing anything, which is good. This is like good things that we like to see. And then we're going to go over the student attitudes. So I looked at 3 different aspects of attitudes. First, perceived usefulness of school science. Do they think the science that they're learning is useful to them in school? And then interest in nature. Do they like animals? Do they like being outside? Do they like having nature hobbies? And then the value of local nature preserves. Do they think wetlands are important? Do they think we should protect wetlands? They think we should protect nature preserves, and all that good stuff. So here is where it gets a little less fun because there's not much of a difference. We can take a look, and they're in their surveys. These are actually all pulled together because all 3 classes did answer these questions the same, because it's just asking them like, how much do you like? I like animals. Agree to disagree and stuff like that. So all 3 of them did take these same questions. It doesn't show much of a difference. Now, this doesn't necessarily mean bad things. There's a couple things that this can tell us. First, it can tell us that field trip is just not changing attitudes. It can also tell us that there's something about seasonality and the time that they are making this field trip that is not really impactful on their attitudes. So all of these field trips were in March and April. March and April, at least one of the teachers was like, oh yeah, our field trip was like on a day that was really cold and rainy. So students could not have enjoyed that. And like, of course, if you are taking a field trip on a cold and rainy day, the next day when I ask you how much do you like being outside, you might say, I don't like being outside. So that makes sense. But it can also give us they can also give us insight into another part. So very early on, I mentioned that if you are younger, you have more positive perceptions of wetlands. In general, the attitudes of wetlands across all these groups are positive. They are 3rd to 5th graders. So what this can tell us is that if we look at some other grade classes, middle school, high school, then we might get a different picture. Okay, and then we're going to look at some teacher results now. Okay, so there are a couple different themes that came up. One, teacher experience. In general, teachers were really happy with the program. Some, they all thought that the program was very organized. Some teachers expressed that they would like some sort of field trip prep lesson or just worksheet to go over expectations for the day, especially because the students are coming back from COVID and not being outside. So it'd be really nice to have something that tells them about the place, about the significance, what to expect, and why we care and why they're going outside. Those would be nice to have. Teacher attitudes. Teachers that I interviewed were all extremely pro outdoor education and hands-on learning. This is great, but it also speaks to self-selection in my data. So basically, if all the teachers that were really happy about being outside and really endorsed outdoor learning were the teachers that accepted and agreed to be evaluated, then there is a point there's an empty point in the data for teachers who might not have the most positive attitudes or the best experiences. So for future research, it would be a good idea to try to get more teachers that are not as happy about the whole process to agree to being evaluated. And then we get to student behaviors. Teachers did mention some improvements of the students' critical thinking and teamwork. They were not really sure if it had one-to-one connection with the field trips, though. One-to-one connection with field trips, they did mention that some students seem to recall information from the field trip as they were doing other lessons. So they would say stuff to each other like, oh my God, it's like that thing we did during the field trip. And then they would like work it out together. So they would recall the field trip often, and they talked about it for a while, and they expressed that they wanted to go on more field trips like it. Lastly, there were just a couple of things that got brought up organically by teachers during the interview that I didn't specifically ask or prompt about. First, it was about students' reading levels. So I had made the surveys to be at a 3rd to 5th grade reading level, and the teachers warned me that the students in general are now, in general, because of COVID, a couple of reading grades behind. So that is definitely something to keep in mind if we are doing any further evaluations in the future. Just keep in mind like general world events and how it impacts students' reading and just student life and existence in general. It was also brought up that there are a lot of students that are English as a second language speakers. So it would be good to keep that in mind in general for those students, even when things like COVID haven't happened. And then additionally, parent participation was brought up. Teachers mentioned that it's getting harder and harder every year to get parents to volunteer as chaperones for field trips. It might be a good idea to have conversations with parents and teachers to see if Yolo Basin Foundation can in any way help motivate parents to join field trips in the future. And I just thought to share a couple quotes from the interviews. So one teacher said, I think it's good for the kids to get outside in nature. When I was a kid, I was always outside playing in the dirt. You know, I like that. And the teacher said, I felt like they, referring to Yolo Basin Foundation, were very organized and very just accommodating and wanting to help. Another teacher said, I saw improvement, especially during experiments. They were really thinking and problem solving. So this teacher also brought up that at one point, one of the experiments they were doing in class failed. And then the students together were like, you remember when that one experiment that we were doing in the field trip failed, and then how we fixed it? Let's try to fix this one. So they just took initiative and tried to fix things. So that's very nice to hear. All right, quick discussion points. So this is a pilot study, so it's wonderful because we can take all we've learned and take it back to the literature, confer with the literature, and tweak things. So we can change things that didn't work, we can add new things, and that's part of something that we can do in the future. Second, scaling. So after we do all of those things, we need to think about how to get more data. And that means we need more classrooms, more teachers, more field trips in different seasons, more ages, and more stations, more of everything in general. So that would be great. But again, with more groups, it means more manpower and more planning. So it's going to take more time, more people, more of everything. And then once we have all of that, we can think about experimenting. So when we get more data and discover more patterns, we can think about manipulating some aspects of the program. So for example, if we are unsure about a specific station during the field trip, we can make an experiment where we update that station for half of the groups that take that station and don't update it for the other half of the groups that take it, and then compare the results. Does it have any effects? And that is all I have for you guys tonight. So feel free to send me your questions now.
Thank you, Sarah. It's really fun for me to see your outcomes. I hadn't had a chance to do that. And these are the kids I work with. So pretty exciting.
Yeah.
Not seeing questions coming in for you. So either guessing you did a pretty amazing job there. Oh, parent participation coming. I'm not sure what you mean, Ken.
Can I talk?
Yeah.
Okay. Thinking back to when I was a kid, I was an outlier being interested in nature, and both parents working, they really didn't have time for that. I think that that is, I think, a constant today with parents having to, both parents working and then take care of the house of their kids. I don't have the answer for parent participation. When Yolo Basin had a school program, we got very little parent participation. And the only sign of success was we had something outside of the school program for kids. And this kid talked to his dad, non-English speaking dad, brought him to the whatever we were doing. I can't remember now, the event. So it's reaching the kids. And, looking at your surveys, you reached a lot of kids in terms of the learning and assimilating the information. I think that's the key. And I think the kid could better convince their parents to participate more than I think the teacher can. So anyway, I don't know. Do you have any ideas on parent participation, increasing it?
Yeah. So one of the teachers brought up that at the end of their year open house, they put up a bunch of the pictures that they took that day with the kids. They printed it out and hung them up, and the kids were super, super excited to grab their parents' hand and like pull them to that like display of the photos and be like, that's where we went. It was super fun. And the teacher put out brochures for Yolo Basin Foundation next to it, and the parents took them all. So recruiting the teachers into doing a lot of the work for us is going to be great. I think it can help. And then getting the students to just talk to their parents about it, just giving them excuses to talk to their parents about it. So if you have places where they can like take, I don't know, Polaroid pictures and take them home with them to show their parents, that might be something. Because they were super excited to show people their photos. And these are young kids, so they don't really have their own like phone or camera with them, but they really wanted to show the photos. So that might be one. And then just again recruiting the teachers into there are teachers that are very loyal to the program, have been doing the program for like 10 plus years and they love it, so they promote it, but getting other teachers to do so would be helpful too.
Great. And last question, do you think there are anthropomorphic factors as to why adults dislike wetlands versus children? And do you have ideas of how we could shift that perspective?
There doesn't seem to be like really specific, and like this is not something that I looked into like super deeply as to why there seems to be a difference. It just seems to be one. But for the most part, it seems developed, not innate, so not like evolutionary, like you're just afraid of water or wetlands. It's because younger students, when they like do experiments of like showing wetlands or not showing wetlands, and then ask them questions about wetlands, they don't really show a difference versus adults do. So there seems to be something going on as we like consume media. There seems to be a really negative association with fiction and wetlands, just getting swallowed by like marshes and stuff like that, and getting sick, and like it's like, oh, it's like they have mosquitoes, and like just following the mosquito talks, they have mosquitoes and diseases. So there seems to be more of a fiction. It comes from stories, not necessarily from like experiences, direct experiences.
Super. Well, thank you. All right, we are going to shift over here. Shoot, where'd he go? Stephen, I just had you. There you are. And there's Stephen. All right.
This one. Haha, got it on the first try.
You did. Nice job.
Okay, so good evening, everyone. My name is Stephen Gergany, and I am a master's student representing California State University, Sacramento. You know, everybody else is from Davis here, I see. And my interest is on the distribution of the giant garter snake and northwestern pond turtle within the lower Yolo bypass, aka the Yolo bypass wildlife area, pre and post flood events. And this is the second year that I have received funding from the Yolo Basin Foundation, so thank you for that. For those of you that are keeping score at home, last year my study was based on these two species distribution during drought conditions. And so I got 1 year of that, and I was going to compare it to the next year. And luckily for us, not luckily for my study, I don't have 2 years of drought. So I will just do a before and after we got a nice flushing of the bypass. So again, the target species that I was looking into are giant garter snakes and northwestern pond turtles. They're both found within the Yolo bypass. And a little bit about them. The giant garter snake is a large garter snake that is endemic to California. So they pretty much only live here. They historically were throughout the Central Valley. Since they're a garter snake, they're non-venomous. They're an aquatic predator, which means they hunt in and from the water, taking prey like frogs and tadpoles and occasional fish. And they're active, during the day and at night if it's warm. And you can usually find them out. Well, they're active from April to October. They are fairly cryptic. But what's interesting about them is that they are classified as a threatened species under the California Endangered Species Act, and also classified as threatened under the Federal Endangered Species Act. So here's a distribution, a historical distribution map of these this species, and if you'll notice, they used to be found again all through the Central Valley, where historically there were the marshes and the wetlands and that. But as we've drained those to do agriculture or put cities there or whatever, their populations have ranges of decline to the point where there's pretty just two population areas. The one up north in the northern Sacramento Valley, and then there's another disjunct population down here in the San Joaquin Valley, and their typical habitat during the active season are canals, rice paddies, open water. They like having some open water with areas to duck and cover in, like this primrose, but they don't like it so much to be completely covered up because it's hard for them to move through it and hunt at the same time. So this is typically, what's found out on the Yolo bypass wildlife area. In fact, it's probably a familiar picture to some people. Again, like I said, they are a cryptic species, so usually, you don't see them, and then when you do, it's just maybe a little head sticking out or a muddy snake here or this. I guarantee you there is a snake in there. So they are not easy to find, so they're not easy to count. And then they overwinter. They do a process called brumination, which is, it's basically hibernation for reptiles. But they move out of the water and into places that like this riprap here or in uplands, and they find burrows to overwinter and hopefully avoid getting flooded out, because whenever they are bruminating, they have reduced their metabolism to really, really low, and so it's really hard for them to wake up quickly. And so if all of a sudden their hole is being filled in with floodwaters, then they have to figure out a way to get out of there, wake up and get out of there quickly. And then the other species that I'm interested in is the northwestern pond turtle. And this is a West Coast endemic species. You can find them in California and Oregon and Washington. They are a thoroughly aquatic turtle, which means that they usually find them up on the ground, traveling anywhere other than for building nests or moving from a dried up area to another wet area. But they spend most of their time in the ponds or on the areas on the banks or basking in the middle. They're interesting. They're important because they are a California species of special concern, which means that Fish and Wildlife is keeping a close eye on them. They have been declining in a lot of their northern reaches for quite a while now, and so there is a push to get a little bit more information on their distribution and some of their habits. And so here's kind of sort of the range that we're dealing with. Up until recently, the turtle was considered to be one species, but they have been split into two. And so the ones that are in the Yolo bypass, which is right around in here, are the northwestern pond turtle. And as you can see, they go northern So this is one in situ, just hanging out by Turtle Pond. And so one of the reasons why I chose the Yolo bypass, beyond just the fellowship opportunity, was the fact that this entire bypass was designed, as probably all of you know, as a big floodwater control, and what it's done is it acts like a natural floodplain that would have existed, out here in and around the Sacramento River, but we have shunted it all into this area. So of all of the surrounding area, the Yolo bypass still somewhat follows the flood up and draw down that the Sacramento's river in this area would have been doing historically, which these species have evolved to have evolved in. And so they're drawn here, they do well here. But my question came about was they're here, and so are they able to escape these big flood events, since they're trapped between these two levee systems, or do they just get flushed out into the delta, and then the population that's up north, do they just get flushed in? So are we just cycling through for turtles and snakes, or are they being able to get out of the way? And so my objectives were to determine the distribution change of the giant garter snake and the northwestern pond turtle within the Yolo bypass wildlife area after a flood event as compared to their distribution during the previous year's drought condition. So I kind of got, both ends of the spectrum on this. And then, with that, I'd like to be able to offer habitat management suggestions to accommodate each species during both flood and drought conditions, pending what data I come up with. So there's different things that I figured could possibly happen here. Basically, I could find them in certain areas, and then in the drought times, and then the next year after a flood condition, I will find them further south as potentially the water pushed them further south into the area. Or I could find them where one year, and then the waters come through, and then find out that they seem to be in the exact same place as before. Or I find them, and then the next year I don't find them after the flood event, or I don't find them the first year, and then the next year all of a sudden they're there. So there's all sorts of potential ways that this could go, and so I needed to see if I could first find them and then find out what they were doing the next year. So first I looked at past distribution studies that have been done on these species and in the general area. And so what I found out was in 2001 to 2012, a wildlife biologist by the name of Eric Hansen did a whole bunch of studies kind of in the wildlife area and to the north. And all the little black dots on here are places that he had study sites, and that he found at least one snake. And then in 2009, or up to 2009, I was able to query all of the distribution of giant garter snakes that were reported to the California Natural Diversity Database, which is exactly what it says, that California Department of Fish and Wildlife curates for people to put in information on rare and endangered species observations, so that they can kind of track them better. So as you notice, they are to the north of the bypass area, both inside and outside of the levee system, but they are up there. And then Mr. Hansen did find them inside the wildlife area right up here against the levee in what I believe is called Willow Slough. So I also did look through the database for northwestern pond turtle observations in the area, and those seem to be a lot less. Oh, here we go. Those seem to be a lot less distributed, or a lot less entered into the CNDB database. In fact, there's none in here. I did it all the way through 2022. I know for a fact that I've entered about 24 of them in this general area, so I think they're just kind of behind on updating that, which is fine. I'm sure they get thousands of entries for everything. But so my interest is that they're not marked in here, and I do know they're there, and so I'd like to get some data, other than just observational, to be able to expand on that. So my sampling locations within the site, I wanted to look at areas that had the good habitat for both species. So a lot of open water with some deep areas and basking areas for the turtles, and some areas to hide for the snake. And so I went through and drove around on the wildlife area and looked for some places, and found a lot of potential areas. And these were set up during the drought times, and so I was kind of trying to find areas that had water in them, because of the sampling techniques, which I'll get to. I required actually having water to be able to sample for them. And so even though there are some ponded areas during more wet season, a lot of these places had little to no water to speak of. And so that's why a lot of this was further over here. And then I did avoid the rice fields, because at that time, and because they were still working, were never technically in drought, because they were constantly this area here stayed the same. So it wasn't my initial, like I said, my initial study was to check drought conditions, so I avoided that area because it wasn't a drought condition. And so the survey method I used was called eDNA, or environmental DNA. And what that is I took samples of water at all of my location sites, filtered it through a really fine filter, and then submitted it to a lab that then checked to see if there were DNA fragments that are unique to the giant garter snake or the western pond turtle inside of that, inside of that filter. And so that's usually from tissue, skin and scales, metabolic waste, things like that the animals have sloughed off as they've moved through the environment. And the benefit of that is all I have to do is be able to take water where a snake or turtle has been recently, a couple of weeks, depending on what the water's doing. If it's just sitting there, that's one thing. If it's flowing, then I kind of have to get a little bit more lucky, but it means that I don't actually have to find a very cryptic snake or a very sneaky turtle. Plus, both of those species require a scientific collecting permit to actually handle. And trap for, and a lot of other bureaucratic things at the university level that would have made this fairly untenable. But there's nothing wrong with taking their bathwater, so to speak, and checking for their presence in that. And so that's why I chose this method. So field samplings, what I would do is I would go out and I would collect some water, usually with a little pole, a cup, and scoop some up and put it in a bucket. And then I'll show you the, or, and then these are some examples of my collection site. So, depending again how the flow was on this, I, might have been lucky, this less of a flow. So, that snake could have come by, 2 days before, and that DNA should still be around in the water. As it got later in the season, my sample sites kind of sort of started drying up. And so I would have a little tiny puddle here, or this was actually just an irrigation ditch way at the south end of the wildlife area that I added last minute because why not? Because it was the only water out there, and lo and behold, I got a snake presence out of that one. But it's not a standard way, but I think it's just because that's the only water that was available to them. So what I would do is I would take the sample and put it in a filter and pass it through, pump it through with a drill bit and a peristaltic pump, and then it would clog up, and everything in here is what I would send to the lab to test. And so again, they would look to see if any pieces of DNA were in there that would have been, unique to a giant garter snake or unique to a western pond turtle. And the technique that the lab used was quantitative PCR, and it looks for genetic markers from specific species in the sample or copied from any DNA fragments that are present and analyzed, which is, again, they're just looking for the pieces of DNA that only giant garter snakes have or only western pond turtles have. And so in this particular study, I got 2, and then I had the bathwater from a stunt snake from another site completely, because I was getting nothing for the longest time, and lo and behold, so I was just making sure that my techniques were correct. So I did end up finding snakes and turtles at multiple sites on the first year. So 2 sites in August, and then the next year I found them at 3 different sites, 2 of which were persistent and one was, by itself. So I did find that they were there. Now, what I did notice is that the first year, this is during the drought conditions, I had snakes here, and I had snakes here. And that was during drought. And then the next year, after the flood, I found snakes in the very same place. And this one right here is one of the locations that is historically known to have a consistent population of them. This one down here and this one over here, not so much, but again, this one here and this one here was during drought, and then the flood waters came through, and I still have them in the historically known areas. And then I have a little bit pushed south here and a little bit pushed south here. So I'm wondering if some of the snakes out of here got pushed further down, or if I just got lucky and found them here and this year, because this is Poudre Creek, so who knows what happens here, but this is a pond. So again, it's just kind of curious on to what my data is going to tell me once I kind of dig down into it. So what I've learned so far is there are giant garter snakes and northwestern pond turtles present in the study area. Giant garter snakes to be more detectable later in their active season, while the northwestern pond turtles appear to be more detectable earlier in their active season. There are giant garter snakes along the western edge of the Yolo Bypass in the Willow Slough adjacent to the levee, and this is a historically known population where they appear to persist. And they were detected in the southern and northern areas. North and south of Poudre Creek was kind of my midline both years. So it seems to me like they're there, and the water doesn't seem to push them out as I suspected. And then spatially, both species don't appear to have shifted. There are more detections of both species after the flood event. So that was interesting, because I was testing in an area where there was less aquatic resources for them to use. So I expected to actually have more hits the year before, because there was less ponds or canals for them to hide in. But and then one interesting thing is that neither of the species were detected outside of the levee. And I checked in kind of right on the other side of the levee where they historically were found in the past. So but I only had that one spot. So that was only 6 samples. So, maybe next time or further, I'll just have to check more outside. And then, of course, the habitat appears to support both species even through a long-term drought, which is good, because I was finding them during drought and they're still there after a flood event. So they survived being restricted by their water resources, and they survived being flushed, it seems. Some management considerations on this. Ensure that the surveyed locations and surrounding areas with positive detections continue to have good aquatic habitat. So if the wildlife area management decides they need to take some water away from somewhere to put it somewhere else, maybe they could not choose these areas. And then, or even provide more open water with positive detections. Because like all but one of the giant garter snakes detections was in an area with ample open water, and sometimes that primrose overgrows a little bit and like force them out. It'd be interesting to see if there is an updated census of the more northern area, like above the causeway, to see if there's some up there. Utilize some eDNA to scan for any potential pathogens, such as snake fungal disease or the shell lesions that are often found in turtles in the same area that these snakes and turtles are in, to see if they might be being affected by that. Because again, I didn't catch any of them, and so I couldn't see their condition. And then enhance the habitat outside the levee to provide a safe haven during extreme flooding events if they have to kind of hop over and out for a while and hop back. So, and questions, comments, complaints?
So we have one. It's about distribution. Is the flood event the only variable or just the primary variable to presence in on the study site?
So it's the main variable that I was using. Again, I had to come up with something to do on year 2, because it was supposed to be 2 years of where are they living while the area is in under drought condition. And so at the general yeah, I guess that is my main variable. I'm going to go back through and kind of do some qualitative studies on the survey locations that I chose to see if maybe there is also other um, upland aspects that might have also affected their presence or absence. But initially it was, is there water out here in the middle of nowhere? And if so, is that water supporting a garter snake or a turtle? And so
Great.
just needed to jump on something else since I already had just 1 year of data.
Super. Thank you, Stephen.
Mhm.
All right. So we're going to come over now to hear from Lauren. Oh, Stephen, if you could go ahead and bring your screen down.
Where is that? Yes. There it is.
There. All right. Hi, Lauren.
Hi, thank you. I'm really excited to be sharing my master's research with y'all. I'm going to make sure that this gets pulled up okay. And, oh gosh, here we go. Dun, dun, dun. Okay, fabulous. So I'm really excited to share with y'all the work that I was doing during my fellowship. I was doing my master's at UC Davis, and I've since graduated and moved to New Zealand to pursue my PhD. And on that note, I am giving this presentation from a field station in the Southern Alps. So if our Wi-Fi tends to be pretty good, but if my video cuts out, just let me know and I'll turn it off so that we can really get good quality audio and presentation. And I'm really jazzed to tell you more about my study, which looks for evidence of a new salmon population emerging in a modified landscape. Ooh, there we go. So first of all, I wanted to thank everyone who made this research possible, especially our funding agencies like Yolo Basin Foundation, the team in the field who had the super fun job of cutting up smelly, rotten salmon carcasses, and our awesome lab and laser team, plus all of the local managers and stakeholders who care so deeply about the Poudre Creek and Yolo Basin ecosystems. So starting off, fish in the 21st century aren't doing great, and fish like salmon are especially sensitive to climate and anthropogenic changes because of their migrations. Salmon start their lives in freshwater rivers as eggs, then they migrate out to sea as juveniles and live in the ocean for a few years, then they migrate back into freshwater, theoretically returning to the same stream where they hatched, where they lay their own eggs and die. So habitat alterations like dams can really disconnect these migratory routes, and they can even extirpate salmon populations from dammed rivers. This map comes from NOAA, and it shows all of the salmon habitat that's been made inaccessible due to dams in the black, and the blue is the habitat that's still available. In addition, persistent and worsening droughts in California are making water issues and fish issues this much more prominent. To date, there are still several rivers in California with wild Chinook salmon populations. However, there's extensive hatchery management to try to bolster these salmon populations. Though some aspects of hatchery management result in captive-born salmon not navigating their adult migrations correctly, and these fish can get lost in other rivers and can sometimes spawn in the wild alongside wild fish. The research I conducted with my grant from Yolo Basin Foundation looks at Chinook salmon in Poudre Creek, which runs through Yolo Bypass. Historical records suggest that salmon could access Poudre Creek during some rainy years, but all salmon access was completely eliminated following dam installations in 1957. In the period after the dams went in, Poudre Creek also experienced habitat degradation. It was even used as the town dump at some points, and a modified landscape emerged in this waterway with urban centers and extensive farming. The creek's connection to the Sacramento was also cut off, and so fish passage for salmon spawning grounds that are located below the dams, which are these blue lines in this red box, also were not feasible during this time. But community concern for the ecosystem culminated in habitat restoration efforts and the implementation of a functional flow regime starting in 2000. A functional flow regime is a restoration approach that aims to mitigate the impacts of dams by ensuring that the water that dams release has ecologically relevant components for the rest of the river downstream, like year-round minimum water amounts, and seasonal pulse flows that can attract in migrating adult salmon and also give a boost to baby salmon that are out migrating. Together, these actions restored quality fish habitat, food, and waterway connectivity. So by 2014, Chinook salmon, both spawning adults and out migrating juveniles, were regularly sighted in Poudre Creek for the first time since the 1950s. The presence of Chinook salmon in Poudre Creek has been hailed as a local conservation success story, but there are still so many questions that remain unanswered about the salmon population, and what it can teach us about how fish can use new habitats in a changing world. We were interested to understand where the spawning salmon that are using Poudre Creek are coming from, if they're primarily composed of hatchery or wild origin fish that are straying in from other rivers. We asked this question to try to identify if any of the adult fish that migrate into Poudre Creek are natal to Poudre Creek, meaning that they hatched here as eggs, then made it all the way back to Poudre Creek themselves as adults to spawn. And if there's yet any evidence of a Poudre Creek population of salmon beginning to emerge after restoration. We were also interested to understand how the development of a salmon population here at Poudre Creek can provide some conservation lessons that can be applied more broadly. So our study approach used really special tools to try to answer these questions. We used otoliths, which are technically means ear stones. They're these little bones inside a fish's brain that grow throughout a fish's life. And the layers of an otolith can tell you information about what a fish was doing during the period that layer was growing. And so we use these layers to track each fish's migration path throughout its life. And the isotopic signatures can tell us about which river they were rearing in as a baby, their out migration to the ocean, and their time in the ocean as an adult. The work on salmon at Poudre Creek is part of a long-term monitoring project at UC Davis, and it consists of annual salmon monitoring crews that float Poudre Creek by canoe every week during the salmon migration season. We count live salmon and we collect samples from salmon that died naturally after they spawned, including the otoliths that were used in this study. We analyzed the otolith's microchemistry. We shot a laser beam along the full length of the otolith from this side to this side, and it gave us data that looked kind of like this. What we're seeing here are ratios of isotopes of one element, strontium, which vary among different waterways due to differences in underlying geology, and they give rivers in the ocean very specific strontium isotope signals. When you're looking at this, these two halves of the graph are mirror images from here to here. And so if we start in the middle here, this is like the very beginning of a salmon's life. When it was just emerging from its egg, it's influenced by its mom. Here is the baby salmon's freshwater residence in the river. Here's its out migration to the ocean, and here's its adult residence in the ocean. And most importantly, we can distinguish different natal rivers and hatcheries where salmon were born based on these strontium signals. If we compare these two graphs, if you look at this signal versus this signal, it suggests that these two fish were born in different rivers. We matched the strontium signals in the natal regions of each otolith to the different isotope signals of different Central Valley rivers and fish hatcheries to tell us how many of our spawner fish were born in the wild versus born in hatcheries. All of these known strontium isotope values on the bottom are the work of really awesome scientists like Malte Wilmes and Anna Sturek, who've been working for a long time in this system. So we used an algorithm to make the highest probability prediction for each fish's natal origin from this isotope data. So for example, for this fish, we identified their natal strontium signal. Then the algorithm matched the value to the best fitting river fish hatchery to find that this particular fish was born at the Coleman National Fish Hatchery, but it died in Poudre Creek. So you might be thinking that using this strontium work, we'll be able to identify any Poudre Creek salmon with 100% certainty, case closed. And while that might be the case for some of the rivers and hatcheries in California that have really distinct strontium isotope signatures, with Poudre Creek, we run into a bit of an issue, mainly that the strontium signal for Poudre Creek is completely overlapped by the strontium signal for the Feather River. This isotope would mean, this overlap would mean that any fish, that really were from Poudre Creek wouldn't come up in our algorithm as Poudre Creek fish. They come up as mystery fish instead. But we wondered if this ambiguity between these two rivers could be resolved if we added more microchemistry methods to help distinguish these two rivers and tell us more confidently if there were any Poudre Creek origin fish. So the other microchemistry method we added to the study uses trace elements. We acquired otoliths from juvenile salmon that had died in their rivers, unfortunately, like this little guy here. And these provided references for us. This time we analyzed a suite of 7 isotopes, not just strontium, to try to identify a specific trace element fingerprint that would distinguish our known Poudre Creek juveniles from our known Feather River juveniles. And once we identify these distinct fingerprints, we can compare the values from the juveniles against our mystery fish to identify whether any of those adults were Feather River or Poudre Creek origin. Okay, so moving into our results, using our strontium isotopes approach, we identified more than 300 salmon over 5 years and found that they came from a total of 16 origins, 11 rivers, and 5 hatcheries. And among these fish, there were 14 that were identified as being of either Poudre Creek or Feather River origin, with overlap causing that uncertain classification. So we move forward in the trace elements work with these 14 fish to try to resolve that ambiguity and identify with certainty if any of these 14 could be Poudre Creek fish. Using trace elements, we identified 2 in the suite that we tested, which did a really good job at distinguishing our known juveniles. They were barium decalcium and strontium decalcium. You can really tell that the Feather River origin juveniles and the Poudre Creek origin juveniles have pretty little overlap, so it's a really distinct fingerprint. The other elements we tested, they had a lot of overlap, so they wouldn't help us distinguish any unknown origin fish. And then moving forward with our 2 chosen fingerprints, we could classify our mystery adults as either Feather River or Poudre Creek origin fish, and 13 of the 14 unknown fish were assigned Poudre Creek, with the last one of an unknown classification again. You can see when we plot our mystery fish against our known juveniles, all the mystery fish in green are much closer to the known Poudre Creek origin juveniles, which are in the blue. And notably, none of the mystery adult fish clustered with the Feather River origin juveniles, which gives us some confidence that these unknown adults had a much closer trace element signature to Poudre Creek than to Feather River. So by combining the strontium and trace elements microchemistry analyses, we were able to identify the natal origins for our 5 cohorts of spawning salmon at Poudre Creek. This graph represents the same data twice. The top panel is just the number of fish every year for each natal origin. And the bottom panel is that same information condensed into proportions. Most excitingly, we detected Poudre Creek origin fish in 3 of the 5 cohorts that we studied, 2017, 2020, and 2021. And we identified the highest number of Poudre Creek origin fish at 11 individuals in our most recent study year, 2021. However, it's really important to note that hatchery origin fish in the purple are by far the most well-represented natal origin classification across all 5 of these cohorts. So to conclude, our study has identified Poudre Creek origin Chinook salmon that were spawning in Poudre Creek, which is a system that's only been supporting salmon consistently within the last 10 years or so. And promisingly, there were 11 Poudre Creek origin salmon in our most recent spawning cohort. We did find that hatchery origin fish dominate in the spawning cohorts in the system, though having so many hatchery origin fish in the first few cohorts could be a good thing to help make a sizable founding population. We also found that the sources of these fish were varied, coming from 11 rivers and 5 hatcheries. And on a methodological note, we found that by combining strontium and trace elements approaches, we could identify our Poudre Creek fish with much more certainty. Our study indicates that salmon are capable of utilizing the novel habitats that are created through restoration and implementing functional flow regimes. And they can begin to develop new populations in places like Poudre Creek. And this information will be really valuable as other waterways, both in California and worldwide, are moving to restore their own freshwater ecosystems. And in some cases, even remove dams so that they can restore waterway connectivity and populations of fish like salmon. So thank you so much. And are there any questions?
That's pretty fun. We've had salmon presentations over the years here and there, and it's really nice to bring it home, you know, to our creek right here.
Yeah, it really brings it full circle, and it's quite fun to have a little bit of a detective story and have an answer at the end of it.
Yeah, absolutely. So first question is, did you think about using eye lenses for run identification?
Yes, we do have eye lenses in storage. We haven't gone around to analyzing them yet, but that is the next goal because we assume that all the fish in the system are full run. However, we haven't determined that using isotopes yet. So it'd be really interesting to see whether that assumption holds true.
And then a comment here, great study. Is it published?
But we are working on getting it submitted to publish very soon.
Excellent.
Yeah, so that'll be coming hopefully in the next year. So it's hard to tell with publications, unfortunately.
Wow.
Mhm.
And you're
But
in New Zealand right now.
I am. It's a little bit after 5 o'clock on Friday.
Wow. AM, PM.
Yeah. Oh, PM.
Well, there you go.
Yeah, it's great. Works out really well with the California time zones, actually.
Well, thanks so much for joining us. And thanks to everybody for joining us this evening, presenting your work. So many great projects have taken place over the years. Oh, got one, Robin's daughter. Please send us a copy of the published article. So
Absolutely.
yeah,
Absolutely.
very fun. So again, I want to thank everyone for coming this evening, learning more about what's going on and all the research that has been done. And a reminder that next month, we're going to be hearing from Martha Diepenbrock about public access and education capital improvement project of the Yolo Bypass Wildlife Area. So that's quite a mouthful, but we're really hoping to get some input as we move forward on this super exciting project that we'll be undertaking over the next couple of years. So thanks for joining us this evening, and we will see you next month.